Welcome, dear students, to this video lecture for Semester 2, First Year B.Sc Microbiology, Course 2, Module 1. The first topic in the syllabus for Course 2, Module 1 is Air Microbiology. Let me give a brief introduction to this paper first, the course which you will be learning in this semester for Course 2. It's more about environmental aspects, about soil, air, microbiology, followed by a separate module on water, that is wastewater and potable water, the different techniques which could be employed for the study of wastewater and so on. In this video lecture, the first topic which I will be discussing and presenting you is based on air microbiology and further aspects of air microbiology. So let us first see what is the learning outcomes for this. At the end of the lecture, the learners will be able to describe the diversity of microbes in air. And the second thing is you all will be able to enlist different techniques to enumerate microbes in air. So let me first begin with the significance of this topic, how this topic will help you. When you speak about the diversity of microbes in air, the first question comes is where do they come from? First of all, make it very clear, air is not a permanent growth medium for the microbes. Air microflora is transient. So you can have a question, justify air microflora is transient. The meaning of the word transient is short-lived. So there are a variety of microorganisms in air. One of the important sources, soil. And now the next question comes, where do they come from in soil? So the natural geological process of weathering of rocks, it is generating soil, and soil has variety of microbes. Soil is a highly heterogeneous system, which you do have a separate topic on soil microbiology, the next topic. So one of the important source of organisms in air is from soil. Now there are various factors which govern air microflora. That is the velocity of wind current, humidity, temperature. All these are factors which govern air microflora. And there are various common airborne infections. That is very important, which you need to keep in mind. The airborne infections could be based on bacteria, fungi. We all have witnessed the time duration for the pandemic that was viral, but that was spreading through air. That's a totally different concept. But the common routine bacterial infections called as pneumoniae and tuberculosis, brucellosis, these are very common this microbial infections which are caused because of the microbes in air. So the next part is, the next question comes is, what is the significance of knowing the techniques to enumerate microbes in air? Now these are all standard techniques based upon samplers which are used to count microbes in air. Now the question comes why and what is the significance of counting? Logically, some of you might be thinking, how can we count something which we cannot even see? These are very tiny microbes which we cannot see with the naked eye. So how do I count? So please make it very clear that especially for the quality assurance and quality control systems in the laboratories, sometimes it's a very mandatory and important technique, an important part of the lab practices where there is a need to regularly monitor the quality of air, especially when you're dealing with sterile fluids and various pharmaceutical drug preparations. They have to regularly monitor the air quality. So there are samplers which will permit a specific volume of air, and depending on the techniques we have important categories. So these are very important techniques which have been in practice for many years. Like the first one is impingement in liquids, impaction on solids, second. Third is filtration, fourth sedimentation, fifth centrifugation, sixth electrostatic precipitation, and seventh is thermal precipitation. So during the lecture in the class, we shall discuss each one in detail, the principle, the working. So this video is basically a kind of an introductory part. The details of each of this technique we will be discussing in the classroom. Now the reference for this topic is Fundamentals of Bacteriology by A.J. Sally. So we shall learn more about this in the lecture. So thank you for listening to this video.